A good morning or afternoon to you. Thank you for joining today's webinar. I'm Jan Reichert, Executive Director of the Antibody Society. This is the second in a series of webinars designed to inform and educate our members and the broader scientific community about topics relating to the adaptive immune receptor repertoire. The Antibody Society is pleased to include a group that works actively in this area. They comprise the Adaptive Immune Receptor Repertoire Community, also called the AIRR Community or AIRR-C. Our speakers are very active and long-standing members of the AIRR community. Drs. Breden, Corrie, and Neller are affiliated with Fraser University in Canada. Dr. Christley is with the University of Texas Southwestern Medical Center. They will talk about the AIRR Community Data Commons, 4 billion reasons to store, analyze, and share antibody, B-cell, and T-cell receptor repertoire data. Please note, this webinar is being recorded. We'll take questions at 2 points in this 90-minute webinar, after the first 2 speakers are finished, and then again at the end of the presentation. Please add any and all questions to the Q&A box in the viewer. We may not get to all the questions, but the speakers have kindly offered to create a Q&A document after the webinar. A link to the document will be sent to all attendees. Without further ado, I'll now turn the show over to Dr. Breden.
Great. Well, thanks, Jan. Thanks for the introduction, and thanks for setting this up, and thanks for helping to run the Antibody Society and welcoming the AIRR community. And as always, thanks to Pam Borgart, who set a lot of this up for the AIRR community. So today's webinar, we're going to introduce the AIRR Data Commons. The first part will be my job to introduce the adaptive immune system in the AIRR community. It basically motivates the whole enterprise. To do that, for some people who aren't as familiar with others with the adaptive immune system, we'll have to talk about the adaptive immune system a little bit. And partly that's to motivate how important I think this enterprise can be, but also to motivate why we need an entire new initiative, an entire new platform for these type of data. They're very unique, they're very complex, they're very hard to deal with, which is also motivating the AIRR community. So I'll be talking about that in part 1. Then Brian Corrie is going to take over and talk about the AIRR Data Commons, more of the details and where we are today, including 4 billion rearrangements that are available through the AIRR Data Commons. And then in part 3, we're going to learn about some of the demos of the tools. Kira Neller, our bioinformatician, is going to talk about the iReceptor Gateway and the AIRR-C data curation. And then Scott Christley will introduce VDJServer as a part of the AIRR Data Commons analysis portal. So part 1, the adaptive immune system and the AIRR community. So as I say, one of the main purposes of this section is to really motivate why a platform specific to AIRR-seq data, and then really motivating the AIRR community. By AIRR-seq data, we're referring to all of the B cells, antibody, and T cell repertoires. Sorry, and I skipped this line there. As I say, the AIRR-seq data, what we refer to as AIRR-seq data, are repertoires of antibody B-cell receptors and T-cell receptors, adaptive immune receptor repertoires. These repertoires have to be incredibly variable to recognize and to remove all the pathogens that we're exposed to during our development, and then, of course, any sort of newly evolving pathogens, such as the novel coronavirus. This system has to recognize and not remove all of the self-proteins in the body, maybe 25,000 self-proteins. And these repertoires are incredibly important to the entire immune system, which brings any sort of disease that has an immune component, such as vaccines or autoimmune diseases or new cancer immunotherapies. So we're working here with the adaptive immune system. And and trying to deal with these AIRR-C data that are so critical to the adaptive immune system. So, as I say, it's important to motivate why you need an entire new initiative for these type of data. And so these next slides are titled AIRR-C data are difficult to share and compare. Partly it's just the size of the data sets. These AIRR-C repertoires have to be highly diverse, as I mentioned, so that they can do their job recognizing all these pathogens, recognizing and not attacking self-proteins. It's been estimated, one estimate is of something like 10 to the 13th potential human B-cell receptors. There's a paper going around that estimates, I think, B-cell receptors as 10 to 61, but that seems impossible. But a good estimate would be as many as in the entire human population, 10 to the 13th potential human B-cell receptors. Now, any individual would only maybe have 10 to the 9th B cells and T cells, maybe 10 to the 6th, 10 to the 7th of those, or not all of those are unique, but many of them are unique. Since 2009, we've been able to deep sequence or use next generation sequencing for these repertoires, and we now sequence 10 to the 6th or 10 to the 7th receptor sequences per sample. And you might have samples before and after a vaccine trial, or inside and outside a tumor, cancer tumor. And so you're producing huge data sets. And even so, with these huge data sets, you're a few orders of magnitude, perhaps, away from describing the entire diversity of these repertoires. But we are producing these huge data sets, and examples here, Burkitt lymphoma, one study had 1.2 billion B-cell receptors. Another one on systemic sclerosis had 700 million B-cell receptors. Many cancer studies every week will have more than 500 million receptor sequences put out into the data commons. So one thing we have to deal with is this huge. Also, these data are highly unique, and this allows me to introduce a little bit of the peculiarities of the adaptive immune system. When we're talking about B cells and T cell receptor repertoires and why they're so diverse, we have to bring in Vdj recombination, which is the first step in producing these diverse repertoires. And this molecular mechanism is unique in all of eukaryote genomes, is unique to the immunoglobulin genes producing antibodies and the T cell receptor genes producing T cell receptors. It's called Vdj recombination. The first step in producing this hugely diverse repertoires are you have sets of V genes, D genes, and J genes in the germline on the chromosomes. And when a B cell lineage or clone and T cell lineage, the original B cell cell or the original T cell develops, the first step is to take one V, one D, and one J and stitch it together to produce the gene that'll be then expressed as the T cell receptor or the B cell receptor. And there's a lot of slides showing this sort of Vdj recombination, but I like this one in that it has a little molecular scissors. There's a little scissors in there that cuts the DNA out between the D and the J, and then cuts it between the V and the D, and produces this Vdj recombination. So again, these are the only genes that have this Vdj recombination, has these hugely expanded families. And this is the first step in the diversity that these repertoires need to have. And partly this unique molecular mechanism leads to the necessity of unique databases with the proper information needed to understand this process, and then unique bioinformatic tools to deal with these huge data sets. And then on top of this Vdj recombination, at these junctions, the enzymes add or subtract nucleotides pretty much at random to increase the diversity. So again, it's a unique mechanism that has to be dealt with. B cells have a further mechanism. When a B cell has a receptor that's recognized as a pathogen, it's told to expand and mutate. And then there's another round of seeing which ones recognize the pathogen those ones that are told to mutate and replicate. And so you have an evolutionary system going on within a B cell repertoire developing as it evolves against the pathogen. So again, it's just a huge amount of variability that's produced by this system. And at this point, I'll mention that, well, clones, when we say clones, we mean the original B cell or T cell that undergoes VDJ recombination, and then all its ancestor cells that develop from that original B cell or T cell. Another reason why these data are so difficult to share and compare and to deal with, there's so many ways for an experiment to differ when a person produces one of these huge data sets. So there's different tissues that can be used as the starting tissue. The sorting can be done very differently. The amplification of the B cell or T cell receptors can be done differently. All of the bioinformatic approaches can vary a little bit between labs. And then finally, definitions of clones or how you analyze these can all differ between labs. So that was one of the real motivations for the AIR community to look at this and say, well, we need to come up with standard protocols and ways to share these data, ways to store these data in a common way. And then finally, they're difficult to share and compare because of the complexity and confidentiality. To make these data worthwhile, you need some information about patient demography, about outcome, about treatment. And so it's possible that AIR-seq data could lead to personal identification. So there's a lot of problems and challenges in dealing with these data, sharing and comparing these data. I'm going to take 2 slides just to discuss how important they can be and how clearly it affects a lot of disease research. This is a slide that also shows kind of what we mean by a clone and how important identifying clones is. So on the left is a sample of CLL, chronic lymphocytic leukemia, blood leukemia. And what you see are these huge expansions. What this is a clone, a B cell, 2 B cell clones that have gone crazy and are cancerous. They're replicating at a much too fast rate. On the right side is a healthy donor. So looking again at the right side, the healthy donor, there's about 12,000 sequences here. Each of the dots represents what our best guess of what a clone is. And we see a very even distribution of sequences among clones. While in the left-hand side, under the disease sample, you have 2 clones totally over-dominating and in a CLL patient. And then the treatment is to try to suppress those clones. And then actually the FDA has one of the uses for these type of data, AIR-seq data, is to try to, once a patient has had these clones suppressed, then the AIR-seq data provide a very fine tool for trying to watch for these clones or any sort of cancerous clones associated to come back. And that's the Adaptive Biotechnologies ClonoSeq test for minimal residual disease. When Brian talks about the AIR Data Commons, he's going to mention our big push in COVID-19 patient AIR-seq data. Here, just to try to talk about the applications of these data, just going to mention some of the trends that have already been seen in these AIR-seq data. And again, what we're doing is making it easy to compare and share these data. So from 20 studies, 3,500 repertoires, over 1 billion sequences from COVID-19 patients, some of the patterns that have been seen, you see stereotypical immune response, you get convergent clonotypes. So different individuals that have been shown have had COVID-19 will have the same sort of B cell clones, the same sequences in their repertoires, showing that they're converging on a particular type of B cell affecting COVID-19 or SARS-CoV-2. We can also see that sometimes these clonotypes are seen in unexposed individuals. So there's a pre-existing immunity. I'm only looking at some of the highlights of these. And for the neuro-COVID, what this paper called neuro-COVID, basically long haulers with neurological defects, Hemming et al. Took 7 studies from the AIR Data Commons, put them together, and showed T-cell expansion in all of the individuals that had these long hauler neurological defects. And then another pattern we've seen a lot, or people have seen a lot through all these data, is very biased V gene usage. This is one of the genes that gets stitched together to produce the receptor. 353 and 366 are V genes that aren't used at a huge amount of in a typical response, but they show up very highly in anti-SARS-CoV-2 antibodies, and they are expressed at very different levels in individuals. So this might suggest that certain vaccines would work better in individuals with certain genotypes, but this type of analysis is just beginning because these data are starting to come in. Point is that we can make it easy to share and compare these data. So finally, this brings us to the adaptive immune receptor community. Basically was formed and motivated to say, okay, how are we going to share these data? Come up with common ways to describe how we produce the data, common formats to store the data. The AIRR community is basically a bunch of immunologists, immunogeneticists, bioinformaticists, experts in IP and consent and legal issues come together to try to learn how to share these data in an interoperable manner. One of the key parts of the AIRR community are the working groups. These meet once or twice a month for years now, and they're very dedicated. Some of the ones that have produced standards, the minimal standards is for publishing and depositing AIRR-seq data. We call it the Meyer Standards. Common Repository Working Group has been working together to produce the AIRR Data Commons. Germline Database has been working really hard to try to understand how we can use these data to understand the germline genes that were used to produce these receptor repertoires. And then finally, also the diagnostics is a very popular working group that is looking at, well, how can we use these AIRR-seq data for diagnostics, partitioning populations, predicting who might respond to a particular novel cancer treatment, things like that. And again, the working groups are the key to the AIRR community. And the way we produce these standards, the working groups work on these standards. They're usually put into the community through a publication, but these publications all have to be ratified by the full AIRR community. So minimal standards produce MyAIR, data represented produce the way of the file sharing. Common Repository produce the AIRR repository web API. And if you're interested, we'll have at the end all these ways of joining the group. But one way to join these working groups is to go to the airrcommunity.org and sign up. So that's an introduction to the AIRR community, why we need this whole initiative, why we're so excited about this whole initiative. I'm now going to turn it over to Brian Corrie, who's going to talk about more details of the AIRR Data Commons.
Thank you, Felix.
Sure.
Yeah, so I'm going to talk about the AIRR Data Commons. And so one of the goals of the AIRR community in the initial paper that was published around at the establishment of the AIRR community was all about making sure that AIRR-seq data was reproducible and reusable. And so that kind of revolves around being fair and trusted. So people have probably heard about the FAIR principles, and maybe fewer people have heard about the trust principles. So FAIR data is all about making data findable, accessible, interoperable, and reusable. And so one of the main goals is to make AIRR-seq data that is fair to the research community. And so what we want to try to establish is a platform and a community that makes data that's easily findable. And through the standards that Felix is talking about, and I'll talk about them in a bit more in a few minutes, making them accessible and interoperable. And really the goal here, and Felix mentioned this, is that really for researchers to really understand what's going on, these data sets are so large, and an individual study has typically a relatively small number of samples from a small number of subjects. And in order to drive new insight, you really do need to understand and reuse other researchers' data. So that reuse of the data is really what drives a lot of the new insights, and we're seeing that a lot more. In particular, the data reuse in the AIRR Data Commons, which I'll talk about in a little bit, has really been driven a lot by COVID-19, and that's it's a pretty interesting kind of what's been going on there. So another important aspect of community and repositories and data reuse. So the trust principles basically are all about data repositories. So not about data sets, but data repositories. And I think we'd all agree that probably reproducible science requires that the data, that you trust the data that you're dealing with. And that revolves a lot around trusting the repositories that store the data. So data needs to be transparent, or the repositories need to be transparent about how they're storing the data. They need to be responsible, have a user focus. They need to be sustainable. They need to be around for a while. The data can't come and go. And they need to be technologically reliable. And trust really revolves around community involvement. And so the AIR community is trying to build up an AIR Data Commons, but in particular an AIR Data Commons that's trusted, right? So people need to have confidence that the data that's in that AIR Data Commons is really reliable. So what is the AIR Data Commons? So the AIR Data Commons is essentially a network of distributed AIR-seq repositories. So as Felix said, these data sets are really, really quite large. It doesn't make sense for those data, all of that data, to live in a single repository. So we believe that a distributed data model is really, really important. Of course, if you have data distributed around the world, it's really, really important that you are able to reuse that data in an interoperable way. And that's really what the AIR standards have allowed us to do. So we wouldn't be able to have an AIR Data Commons if we did not have the AIR standards that we rely on. So right now, Felix mentioned, we have currently 4 repositories, but our vision is that this is going to grow pretty dramatically over the next few years. So we expect tens or maybe even hundreds of repositories. Currently, those 4 repositories that we have now have 60 studies and over 4 billion sequences. And it's important to note that those 4 billion sequences are highly processed sequences. So those sequences have gone through a significant curation process. They've been annotated with the VDJ genes, so they're not just the raw sequence, which is the type of data you would get out of the NCBI SRS. Driver for that is so that you can basically query each of those repositories and do that in a way that is transparent to the end user. So we want people to be able to actually ask the same query or question of each repository that's in the AIR Data Commons and get back a result. And the way we do that, again, is relying on these standards. So the Meyer Standard, Felix mentioned this, it's a standard for study reporting. And it basically has roughly 80 fields that the community recommends that when you're preparing a study, you consider capturing when you're preparing your study. The file formats standards are all about data sharing. So it makes it possible for, if I get data from one repository and I get it from another repository, I can use them together. And then finally, the ADC, the AIR Data Commons Web API, is a web-based query API so that you can send all of these queries out to the repositories in the AIR Data Commons and get results back in a consistent format. Now, one of the key things that, so having data being fair is important, clearly, but it has to be easy to use. So one of the things that we've been working on in our project in particular is what we call the iReceptor Gateway. So the iReceptor Gateway is a web portal. It's designed for interactive web-based discovery, exploration, and analytics of AIR-seq data. And what that does is it basically allows you to issue queries through the web portal, and it hides the complexity of finding and searching and federating data across many repositories. So essentially what the user does is the user sits in front of their web portal, and Kira is going to kind of walk through a demo of this a little bit later. You sit in front of your web browser, you say, I'm interested in cancer studies, you click a button, that query goes out to all the repositories around the world, the data comes back, and you get the results. And so one of our goals is to make it data be findable, accessible, interoperable, and reusable, but to do that in as easy a way as possible. So make it simple for the end user. So why should I use the AIR Data Commons? So the AIRR Data Commons has been growing pretty dramatically. So there's an enormous amount of AIRR-seq data out there. Even though we have 4 billion sequences from 60 studies, that is a small portion of the data that is available. The value of the data is really, really important. As I'll talk about in a bit, we're seeing a lot of data reuse. And so the AIRR community is encouraging everyone to share. And if you do want to share your data, there are many mechanisms to do that, and we'll talk about those in a bit. We encourage people to reach out to the people in the AIRR community and share. There's several mechanisms by which you can do that. So one of the interesting things about the AIRR Data Commons is its growth. So basically for most of 2019, particularly the first half of 2019, the AIRR Data Commons grew very, very dramatically. Kind of as you can see in the graph, in kind of late 2019, early 2020, we were working on the platform. But as we all know, in early to mid 2020, COVID hit, and we saw a dramatic increase in interest in the AIRR Data Commons and the data that was in it. And one of the reasons for that is, of course, that we spent some time curating that COVID-19 data. And one of the interesting things I think that COVID-19 showed about the value of the AIRR Data Commons is that people really valued the data that other people were producing, and they really needed that data to generate some results. So just a little bit of a picture of kind of what happened. In May 2020 was the first preprint that we were aware of that had AIRR-seq data in it from COVID-19 patients. By June 2020, that data was available in the AIRR Data Commons. This is from the preprint from Nielsen et al. And that data was being interactively searched by a number of users almost immediately. That final paper for that preprint came out in October of 2020. So one of the things I mentioned is data reuse is being very, very common now. We're seeing a dramatic increase in researchers finding data in the AIRR Data Commons and reusing that in their further studies. And I think a lot of the data reuse has been the importance of data towards fighting the COVID-19 pandemic. So we've had, we had papers published in May, June, July, August. Those papers were curated into the AIRR Data Commons. In particular, the Schulthess et al. Paper, which many of you may be familiar with, it was published in August of 2020. And we had a researcher download that data in September of 2020 and reuse it and publish another, at least a preprint on that data reuse within a couple months of having that data online. And so that data reuse is becoming very prominent. And I think the value of having data in the AIRR Data Commons and the value to the community as a researchers is quite high. And I think the value to the researchers that are sharing the data, I think, is quite high as well. Their research is being reused in a pretty significant way. So a little bit more on use of the AIRR Data Commons and kind of where things are at. So again, a lot of this driven by COVID, as you can see in the right-hand graph, in July of 2020, so the first data set went online in June 2020. We typically, previous to kind of the COVID data influx, we would get 2 to 3, 4 maybe users a month. So maybe you'd have 20 new users in a good month on the iReceptor platform. When we made the announcement about the COVID-19 data in July of 2020 alone, we had over 70 new users join the iReceptor Gateway and have access to that COVID data. And as you can see, the new users have stayed high ever since that time. In the left-hand graph, you'll see the types of users. So there's a very high interest internationally. North America is very prominent, Europe is prominent, Asia is prominent. One of the interesting things I find is that we have a fairly significant number of commercial users. So we have 52 commercial users from kind of biopharma types of companies over the last since June 2020 as well. So we think this is being driven by two things. Certainly COVID-19 is a big part of it, but I think also there's a critical mass of data now that's available. A lot of that is COVID-19 data, but it is also, there are also other types of data, and Kira is going to talk a little bit about that later on. So I mentioned the iReceptor Gateway. I just want to, and Kira is going to talk about this in more detail, but I just want to make it, just to give a quick example of how easy it is to find data in the AIRR Data Commons. So as I said, you can kind of go to the iReceptor Gateway. You can type in basically a simple disease diagnosis. I'm interested in a typical type of B cell or T cell. Do a quick search and go from the 4 billion sequences that were initially in the AIRR Data Commons to finding only those that are related to COVID-19 studies. So there's 1 billion sequences in the COVID-19 studies. Another capability that you have is to do a quick, simple analyses. So we have the ability to do analyses on individual repertoires or samples in the AIRR Data Commons. You can get V gene usage, D gene usage, J gene usage, junction length, and amino acid usage as well. So these capabilities make it easy to find data of interest, drill down, get that data, and then download it and do further analyses offline. It is possible to do a more kind of computationally related types of analyses. So you can use the AIRR Data Commons API directly. It is a web-based API. So if you are a bioinformatician, you're writing your own code, or you want to search for something from the Unix command line, you have full search and download power for all of the repositories in the AIRR Data Commons from any of those platforms. So I'm going to walk through a simple use case. So the use case is basically, I'm interested in the V gene usage for antibodies that are antigen specific for SARS-CoV-2. So I'm not sure if people are aware, but the Oxford OPIG group has developed a really valuable resource. They have a database that has SARS-CoV-2 specific antibodies. So these are antibodies that are known to be specific to SARS-CoV-2. So in this use case, basically what I'm as a user, I'm interested in looking for a specific CDR3, which is a feature of interest. It's basically prominent in binding between the antibody and the SARS-CoV-2 virus. And so I'm interested in looking for an antibody that's known to be specific. I'm interested in the CDR3 feature, which is basically this amino acid string right here in green. And if I want to query, and you probably don't really need to read the details of this too much, but essentially what I'm doing is I'm sending a request to a specific repository, which is down at the bottom, covid19one.ireceptor.org. I'm looking for the junction AA field, which is basically the amino acid CDR3. And I'm looking for the sequence of interest, which is that amino acid string. And I'm asking it to give me the vcall response to that query. And when I do that, I basically get back the data from the repository that meets my search criteria. So this is a SARS-CoV-2 antibody specific CDR3. And if you'll recall, Felix was talking about how IGHV353 is a gene that's been known to be prominent. And sure enough, we see a pretty high representation of IGHV353 with this search for this CDR3 from this specific antibody. So that's a pretty powerful query to do with just a couple of commands. So another thing that the AIRR Data Commons allows you to do is, of course, download and reuse this data. That is really the main goal. And how do you do that? One of the things that the AIRR community is working on is creating a AIRR software compliance badge. That badge is targeted at software components that have demonstrated that they follow the AIRR standards. So some of the requirements of the badge are that the software is open source. It supports the AIRR standards. It has example data, so you can test it. It has some sort of containerization, so it's easy to manage and install. And it has some sort of user support. So there are a number of tools that have been certified thus far, and they're listed here. And we would encourage people to, if you are a tool developer, to consider looking at the AIRR Data Commons or the AIRR Software Compliance Badge. So far I've been talking about using the AIRR Data Commons to find data. The key things that we want to encourage through this platform is researchers sharing their own data. So the data in the AIRR Data Commons is only as valuable as the data that is provided by the research community. So there's a couple of ways, and if anybody is interested in sharing their AIRR-seq data, if you're running studies, I recommend you reach out to your colleagues in the AIRR community. There's some contact information at the end of this presentation where you can contact various groups around getting help for sharing and publishing your AIRR-seq data. There's a couple of real simple mechanisms to do that. If you are a group that has a lot of AIRR-seq data and you generate a lot of AIRR-seq data in your own studies, you may want to consider running your own repository and having that repository a part of the AIRR Data Commons. If you're interested in that, you can download what we call the iReceptor Turnkey. It is basically a set of Docker containers. It gives you a turnkey database and web service that allows researchers to query your repository. If you want more information, you can go to this link on the screen. These slides will be available after the talk. Another mechanism that you have is working through VDJServer. And Scott's going to talk about this a little bit more as well. So that allows you to upload your data to an existing repository and then publish that data by processing it through the VDJServer pipeline. And again, if you want more information, I'd suggest you reach out to VDJServer, and again, contact details will be provided a little bit later. So I'm almost wrapped up with kind of the high-level overview of the AIRR Data Commons. So there's a couple of things that are coming down the pipe that I think people might be interested in. Right now, currently in the AIRR Data Commons, we have only rearrangements or annotated sequences in our repositories, but there's a couple of other very, very important developments that have been coming down the pipe. So single cell analysis has been really taking off. A lot of the COVID studies that we're seeing are single cell. We currently don't have the capability to store that, but that capability is going to be released in the not too distant future. And Felix talks a fair bit about clones and clonal lineages and how important they are. That is another extension that the AIRR community is working on from its standardization process. And once that standardization process is done, you can expect to see clonal data and clonal lineage data in some of the AIRR-seq repositories. And one of the other things that I just wanted to mention really quickly, so we're a number of us, of the colleagues in the AIRR community are part of a project called the iReceptor Plus project. And one of the focuses of that project is integrating more advanced analysis tools into the platforms themselves. So right now, if you use the AIRR Data Commons, you can find data of value, but you have to download it and run these pipelines externally. So we're working on extensions that allow you to do more complex analyses without having to be a technical guru and know how to run all these software pipelines offline. But these pipelines will be embedded in the actual user interfaces for things like the iReceptor Gateway and VDJServer, so that you never have to leave the web-based user interfaces to do your analyses. So I think we're going to pause there for a quick Q&A session. So we have maybe 4 or 5 minutes to just answer a couple of quick questions, if anybody has any questions.
Hello, everybody. We're back. So we do have a couple of questions. First one is there a range of unique B-cell receptors to particular B-cell subtypes from one individual?
Sounds like a Felix question to me.
Sounds like a Jamie Scott question, but she's not here. Well, so I think if you're talking about different B cell, like the different sorted out B cell populations, there are many of the studies have done that. A lot of them are just bulk sequencing of PBMCs that haven't been sorted, but a lot of the studies have been sorted by B cell subtype and then sequenced and put into I don't know exactly how many right now, but if somebody wanted to contact us, we could show them how to, you can go onto it and ask for naive B cells or, IgG type B cells. So those data are available, and we want to expand in that area. Yeah.
Second question, if we're ready. How does iReceptor ADC compare to the observed antibody space database? Is most of the OAS data likely to also be available through AIRR Data Commons?
Yeah, great question. So OAS, the observed antibody space, that's actually out of the OPIG group, which did the COVABDAB as well. Yeah, so their repository is AIRR compliant in the sense that it has, if you download data from it, you can get the data in an AIRR compliant format. So you could compare it with any data that is in the AIRR Data Commons. What they don't have at the moment is an implementation of the AIRR Data Commons API to query it. And so you can't query it through the gateway, the iReceptor gateway, for example, because we don't have an AIRR Data Commons API implementation for it. And we have been chatting with them off and on about that. So that might be available in the future, but right now you can download data, reuse it that way, but you can't query it interactively through the gateway.
And do we know how much overlap there is between OAS and our 4 billion? I think we've looked at that, but I can't remember off the top of my head.
Yeah, I can't remember off the top of my head, and we haven't looked at it for probably 6 months, so I'm not sure what the current state.
What Felix is. Yeah, I was just going to say, getting these same data sets in different repositories, we're trying to struggle with how to quantify that and warn the users about that.
Yeah. So it's possible to have different annotations with different pipelines for the same source sequence data. And so that is actually quite valuable because you can compare and contrast different analysis approaches. But it also makes life a little bit difficult because you have to understand how the data was processed and realize that there are two different data sets that have the same source data. So it's when you have this network of repositories, it does add certainly some complications around that.
Excellent. Well, thank you very much for answering those questions. Oh, here, do we have something else? Support, question mark. Types of amino acid mods, like modifications. Even I got that one. Like phosphorylation or carbohydrate modifications. Is that cause you
right. If I understand, the database model for the present doesn't take into account those type of modifications. I don't think. But
okay. Well, thanks very much to our first two speakers, and I think now we'll move on to our third speaker.
All right, Kira, over to you.
All right, great. So here I'll be giving just a quick sort of walk through the iReceptor gateway. And as Brian described, this is a web-based user interface for querying the AIRR Data Commons. So when you first navigate to the gateway homepage, you'll see a page that looks like this, we start off with a little bit of a blurb that describes the intention of the gateway. So that is to enable the discovery, analysis, and download of AIR-seq data. You'll also see a panel here that describes some of the features of the currently 60 studies that are in the AIR Data Commons. So you'll see study types, tissues, diagnoses, and here in terms of the diagnoses, you can really see that all the excellent data sharing and collaboration efforts we've had has resulted in COVID-19 being the most abundant diagnosis that we have in terms of representation of studies. So I mentioned that, we're not seeing the slides change.
So I just wanted to confirm, so I'm not seeing the slides change on my end when Kira is talking about the different slides. Can anybody else confirm that really quick? Sorry to interrupt, Kira.
It's okay. Okay, maybe I'll just use the bottom.
So Felix, are you seeing the slides change?
I'm still on. Oh, now I saw a slide change. Yeah, now they're changing.
Okay, so sorry, Kira, go ahead. I'm going to assume that maybe other people are seeing it, and it's a problem.
You're on home page right now.
Yep.
Okay. Continue. Okay. Sorry.
All right. So where was I? Presently, we have 60 studies available in the AIR Data Commons. And by clicking on the studies button, you're able to see all these available studies. They're organized by the different repository. So here I'm just showing a snapshot view. We have the AIR COVID-19 repository, and this is just showing the first few studies available within it, which are actually all from Nolan et al. So this is the big adaptive study that we have for COVID-19. And we split this up into 7 different studies. And by clicking on each title, you'll be brought to the original publication. So back at the homepage, we also have a little What's New panel where we announce new data that has been loaded into the AIR Data Commons, as well as new features that we want to alert you to. So for example, here, the statistics feature. In order to access the gateway, you require a username and password. So you can get these things by applying for an account by emailing support@iReceptor.org. And once you have this information, you can then log in. So you'll see a page that looks like this. And you'll see that there are 2 main search capabilities that you can use once you're in the gateway. So these are the repertoire metadata search and the sequence quick search. So I'll begin first with the repertoire metadata search. And here you can activate this by just clicking on the blue button, Browse Repertoire Metadata. That will bring you to the search results for the metadata search. So here you're seeing all the 4 billion plus sequences from 6,110 repertoires. So this is all the data that's presently in the AIR Data Commons. We provide a default layout that presents some of the key MyAir fields. However, you're welcome to customize this layout by clicking on the Customize button. And then here you'll see you can choose any of the MyAir fields that you want to display. So as I mentioned, what you're seeing here by default is all of the data in the AIR Data Commons. So likely you'll want to filter through this data to find some data of interest. So we provide different mechanisms for that. You can apply the full text search like Brian showed before, just entering COVID-19, you'll pull up all the COVID-19 data sets. You can also filter by study parameters. So title, type, group, subject, for example, the subject ID, the sex, ethnicity, age, important one here is the diagnosis, and sample. So if you want to search by PCR target, so this would be IgH, K, L, TRA, or B, different cell subsets, tissues, phenotypes, etc. And if that is not enough, you can also add a custom filter by clicking on the Add a Filter button. You can click Filter of Interest. So here I've just selected the repertoire description. When you click on that, you'll be able to press the Add Filter button, and then it moves up into the active filter. So in this case, what you'd be able to do is search on any keywords that are within the repertoire description field if you want a bit more control over your search. So here back on the Gateway metadata search results, I've activated the COVID-19 text filter. So what we see is now a billion plus sequences from 3,624 repertoires. So this is all of the COVID data that pops up when we do that search. And at this point, what you may want to do is download the metadata associated with all these repertoires. So you can do that depending on your desired format, so either JSON or TSV. Click on that one of those buttons and it'll download the metadata. You can also explore individual repertoires. So if you want to have sort of get an overview sense of the repertoire before you choose to download the data, you can take advantage of the new stats feature that we offer. So by clicking on the stats button corresponding to a repertoire of interest. So click on the stats button and you'll pull up the stats panel. So here we're showing the V gene distribution for a certain TRB repertoire. You also have available the D gene distribution, J gene distribution, and the junction length distribution in amino acids. So what is shown here is the default view, which is the very top level. So the subgroup or family according to IMGT nomenclature, the top level. However, if you click on any of the bars, you'll be able to drive down into the next level. So for example, clicking on TRB-V19, you'll be able to see the gene distribution of that family, and then clicking once more, you'll be able to see the allele level distribution. So there are multiple levels of sort of granularity that you can look at. So back to our metadata search results. At this point, you've probably identified some repertoires that you are interested in and you want to start looking at their sequences. So you can do so on an individual repertoire by clicking in the sequence column I've indicated here. If you click on the button corresponding to your repertoire of interest, you'll be able to go to a sequence page, or you can browse the sequences from all the 3,624 repertoires. Either way, you'll then activate the sequence search page. So this page comes up. And first I'll note that you have filters, additional filters on the left-hand side. So you can filter by specific V, D, or J gene, and by a junction or CD03 amino acid length. And once you've filtered down your sequences of interest, you can then download all these sequences by clicking on the button indicated. So that wraps up the repertoire metadata search. And now we have to walk through the sequence quick search. So as the name suggests, it is quite quick. All you have to do is type in your junction or CDR3 amino acid sequence of interest. So here I'm using the same one that Brian used in his demo before, when he was querying through using the ADC API. But here we are using it through the quick search. So you just type it in and we get the same results that Brian received. So the 906 sequences, which you can then download using the same download button as mentioned before. So that wraps up the demo of the iReceptor Gateway. Okay, sorry, just got a message. So the iReceptor Gateway is all about finding your data of interest. But at this point, hopefully we have shown to you the value of depositing your data. So to deposit your data, you'll need some tips for data curation. So hopefully with some of these tips, it'll smooth the process out for you. So for curating an AIRR-C study for the AIRR Data Commons, you need two main things. You need your rearrangement annotation files. So this will be one per repertoire in your AIRR-TSV format. For more information on the format, you can follow the link. As well, for the metadata, you'll need one file for the entire study, of course, following the AIRR reporting standards that have been discussed. And again, you can follow the link for more specifics on these standards. So as has been described, the AIRR reporting standards are 6 high-level data sets that have been found by decided from the AIRR community as being critical for publication and data deposition of an AIRR-seq study. So I'm not going to go through all of them here, but this figure is just showing all the individual elements within each category. And it is fair to say that this takes, a little bit of effort to curate these fields. However, I will say that although all fields are important, they're not all required. So not all of these fields are essential in order for you to load your study. However, we do, of course, encourage you to provide all available metadata that you have. And again, for a description of examples of each of these elements, feel free to follow the link indicated. So I also want to point out our iReceptor GitHub. If you want to go there, we provide many resources that can guide you along in your curation. So for example, here I'm showing just a screenshot of the data curation page that we have. We'll have examples for both metadata curation and rearrangement curation. So rearrangements, we provide different file formats. So the AIRR format, mixer format, IMGT-VQuest. But what I think is particularly helpful is the metadata example. So here I'm showing a screenshot of that file. It's basically just an Excel file, which you can download, and you can see we've documented, I think, 5 or so different study types. So this will give you a nice example of the data curation requirements when you're working along on your own. We also thoroughly document our internal data curation process. So if you follow some of the links on this page, you'll be able to see some practical tips we provide in terms of sort of the layout of your data, all the files and folders, and it's pretty well documented, just some practical tips. But of course, if at any point you look very confused, like this math lady meme here, please do reach out to us. So you can email us support@ireceptor.org, or if you have any specific curation questions, feel free to email me as well. So I will end my little portion of the talk here with just some of the common issues that we have encountered, or common pitfalls, and why these have posed a problem. So sort of demonstrating the importance of these issues. So number one being not providing your annotations in an AIRR-TSV format, or a format that can be converted to AIRR-TSV. This simply is an issue because non-AIRR compliant data cannot be loaded into a repository. However, I will note that some of the data loaders do accept output that's directly from different annotation tools. So the ones provided that I've indicated there. So Mixer, IMGT-VQuest, IGBlast, AIRR-TSV, these are some of the standard tools. Only providing process sequences. So for example, if researchers only provide the CDR3s rather than the full input sequence that was actually used for the annotation, this hinders data reuse. So others won't be able to actually take those sequences and, for example, do any other processing that they want. And if others want to verify the original conclusions, having the full raw sequences is important. Like the AIR-TSV format for the annotations, the metadata has specific formatting requirements. So these use the Meyer fields. And if you do not pay attention to the formatting types for different fields, for example, string, ontology, integer, etc., the data loader will not load your data. So be mindful of those. We encourage researchers to provide all data or all processing steps that are available. So the tools and the versions, the quality control steps, and the specific reference genomes that they use in order to generate their data. This is especially important because when a researcher is looking for data sets of interest, they want to know exactly what they're getting. Also, if a researcher wants to, again, do any different processing, they need to know what the current processing is. An important one is not using the same subject or sample identifiers as in the published paper. So this is an issue because then others can't easily compare the data set to the paper. This is important because some users are seeking only, a few repertoires of interest from the whole data set, and they need to be able to find which ones those are. And we're not able to provide them that information if it's not clearly indicated. So we'll usually ask the users to contact the authors directly. And finally, I just want to point out that although your study may not have considered certain variables, for example, maybe you didn't consider age or ethnicity, if you have this data available, we highly encourage you to curate it, just because others may need these data for reuse. And if it's not there, then this might mean that they're not able to use your data. So with that, I'll pass it over to Scott for a VDJServer analysis.
And we hope Scott's there. Oh, it looked like Scott may have just dropped off, so he might be rejoining. Please bear with us for a second.
Hi.
There he is.
Hopefully I'm connected now. Sorry about that. I got frozen. Thank you, Kira. Yeah, so I'm gonna present a few slides on VDJServer. VDJServer is a analysis portal that was developed through NIAID funding here at the University of Texas Southwestern Medical Center. It provides a complete analysis workflow for processing your AIRR-C data and analyzing and visualizing. We'll show in a couple slides, or at the end of my slides, we'll show a short video that introduces some of the interface and the analysis capabilities of VDJServer. I'm giving a list right here. What I'd like to talk about today more specifically is the Community Data Portal. The Community Data Portal is a way for users to publish their studies, make their data, metadata analysis and visualizations available to the public. Now you can also, through VDJServer, just share your data privately with other VDJServer users. But what I'm talking about here today is how to publicly share through the Community Data Portal. It's important to remember that you don't have to actually run your analyses on VDJServer in order to share data. Though if you do, then that will provide more capabilities, and that analysis that you run will be available publicly for other people to look at and visualize the results. So the sharing of data is very easy. It's a 3-step process. You create a project, which is what VDJServer uses as a sort of virtual container for files and analyses. They're all contained within a project. You upload your data files to that project, and then there's a button you push, and that will publish it and make it public. And then anybody would be able to query and download that data. Now that's a facility that VDJServer has had for a number of years. But the new thing, and what we've been talking about today in the AIRR Data Commons, is a new feature that we've been adding to VDJServer as well. Here we're involved in both parts. The first part being actually storing data in a repository. We have a little over 20 studies and about 2.5 billion rearrangement records in our database that you can search through the API. Interestingly, almost all of the studies have been processed from the raw data using VDJServer's analysis pipeline for consistency. So it's the same set of pre-processing, VDJ assignment, and analysis that gets run on this data. Also, the same germline gene set is used for all of the annotations. So besides being a data repository, actually providing some data, we're working on updating the VDJServer graphical interface, version 2 of it, to bring new capabilities. One thing is AIRR data entry for studies. So currently VDJServer does provide metadata entry screens. They are pretty close to being Meyer compliant. They collect almost all of the Meyer data fields. The only thing that's really missing is we don't support the new file formats for holding that metadata, the AIRR repertoire metadata format in particular. Along with that, our integration with the iReceptor Plus platform, and that was mentioned a little bit earlier. So that's a sort of a larger initiative to bring analysis to the AIRR Data Commons and create a lot of the sort of underlying infrastructure that's needed to do that, especially to handle these large data sets. So one of the kind of the key unique features of that VDJServer is that it will allow you to perform comparative analysis between your own private AIRR-seq data that you have in a project and any data that you query from the AIRR Data Commons. So, and was also mentioned before, there's a lot of ongoing effort to standardize beyond right now rearrangements and study metadata, but also additional data types like clones, trees, lineage trees, single cell, and also standardizing how we describe data processing and, most importantly, the germline gene sets. Okay, so publishing your data is easy in VDJServer. It's a little bit more complicated to actually publish a study in the AIRR Data Commons. There's some additional steps. Most importantly of these is that the data needs to be compliant with the AIRR standards. That means the metadata needs to be all in the AIRR repertoire format, and the rearrangements have to be in the AIRR TSV format. So now that would be automatic if you ran your analysis through VDJServer's analysis pipeline. the outputs will all be in AIR standard formats. However, if you upload your data and you don't use the analysis pipeline, it'll be up to you to make sure your data is in those formats. Okay. In terms of moving from version 1 to version 2, if you enter in metadata into the server today, we'll be able to convert that to the new AIR metadata format. We already have scripts and programs and so forth that'll do that conversion. So, and then lastly, to actually put it into the database, you've heard a number of times how big this data is. The actual processing of loading it into the database can take a while. We're talking hours to even days for some of the large studies. So right now we have an administrator that performs that actual last act of doing the database load. But that's pretty easy for us to do. You just sort of contact us when you're ready, or even contacts ahead of time. We can help you through some of the various steps of curating your study, making sure that it's compliant with MyAIR and supports all the AIR standards. So, and then to finish up here, we're going to show you a demo. This walks through, as I say, I didn't talk about very much of the analysis capabilities, but this video, short video of about 2 minutes, will walk you through some of the analysis capabilities and through the graphical interface. So if you would please, Trevor, could you start the video for us? The link is there for people to use. I believe, though, that we should be able to show you the video. Great. Well, thank you very much. That's the end of my session for VDJServer. I think we have a couple slides here to finish up. Brian or Felix, were you going to walk through the last slides?
Yeah, I was going to walk through these last slides. Looks like my camera's okay. And these slides should be available for this information. It's 3 more slides here. There's several websites you can get information from the AIRR community, how to join the working groups. Get links to all of the meetings we've had and YouTube videos from the meetings where we have scientists talking about AIRR-seq data, but also how we produce the working groups and to produce the standards and kind of the governance, and how we take this from a community initiative, kind of a ground up initiative. The iReceptor website at iReceptor.org and the Scientific Gateway. Ryan's mentioned that you can get an account on that very easily, and Scott's gone over VDJServer at VDJServer.org. iReceptor Plus is a large Canadian/EU supported initiative with 19 institutions in 9 countries to expand the AIRR Data Commons and the analysis tools available on the AIRR Data Commons and science using the AIRR Data Commons. And you can get information there at the iReceptor Plus website. Got several GitHub sites for the where we work on the cyber infrastructure and have the discussion groups about that, you know, how to get this done. And there's several publications. Again, thanks to the tabs for helping support these. And again, you can get these publications from the AIRR community website under tabs, but several publications that talk about the different standards and protocols and cyber infrastructure and software, interoperable software and everything. Looking for my next slide. Then the contacts are in this slide for, again, the AIRR community, email addresses for the overall communications, for the data commons, for documentation, for the iReceptor project, and for VDJServer. So all those accesses are there. And like Brian said and Scott said, to get help, you can either run your own repository or get help uploading your data and curating it through our wonderful components of the AIRR Data Commons to really increase the impact of your data and the use for biomedical research. Finally, acknowledgements. The colleagues in the AIRR community have been going for 4 or 5 years. People have worked very hard, very consistently on this. We've had, I think, 5 meetings. We're going to have hopefully an in-person meeting. We're certainly going to have an AIRR community meeting in December, hopefully in person. Collaborators across iReceptor and iReceptor Plus. Funders, iReceptor Canada has been funded by Canary, CFI, CIHR, BC Knowledge Fund, VDJServer, NIAID. And I mentioned the iReceptor Plus as the EU Horizon 2020 program and CIHR. Platform providers have been Compute Canada and the Texas Advanced Computing Center. And I think we have some time for a few more questions if I guess Jan is our question person. Hey, Jan.
I'm back. Well, thank you very much. It does appear that you all were such fabulous presenters. You did such a fantastic job explaining everything. Every slide was spot on in terms of exactly what you needed to say to keep people informed and educated about what you are doing. Thus, I am getting to the point of saying we have no more questions.
Everybody.
I think everybody's just excited about, you know, adding to the AIRR Data Commons. I think we've had 5 new account requests during the presentation. So.
Well, that's fantastic, and you should have all 55 or more plus people who attended definitely signing up for accounts and getting active with your activities. Any last words before I conclude?
Well, just to mention, I mean, even if you don't think you're going to use the data in the Commons, it's fun to just get an account and to start looking around at how you can, you know, search what's out there.
Yeah, Felix, the other thing I'd maybe also just say is, you know, the AIRR community is very open. If people are interested in participating, we're always looking for new members in the working groups. So if people are interested in participating, there's lots of opportunities to do that. reach out to the AIR community email that was on the previous slide.
And the slides are available to you to download. They'll also be on the Antibody Society's website very soon. Any other comments? Okay, with that,
Thanks for everybody's time.
Yeah, I'd like to thank you, well, not only to the attendees, but also the speakers. So I'd like to thank our speakers for relating their insights and experience with the AIR Community Data Commons. I also want to thank everybody for joining the webinar today. An on-demand version will be available in a few days, so I'll send a link to the on-demand version to everybody that registered, and I'll also send a link to the Q&A documents so that you also have that available. Please do feel free to watch this or any of our on-demand webinars when it's convenient. And I thank you again and have a great rest of your day.
Thanks, Jan.
Bye. Thanks to Tabs. Bye.
Thank you.